Okay, we shall continue with the lecture on infectious diseases. Okay, so if you recall from the previous lecture, we learned about B cells. We learned how B cells form antibodies, right? The antibodies, whether they are membrane bound as B cell receptors or they are secreted, right, as antibody proteins. Regardless, right, we learned that the B cells produce specific antibodies, right? So a single B cell would only produce antibodies that is specific to one epitope. And different antibodies, right, specific for different epitopes would be formed by different B cells. Okay, so how does the B cell, right, come up with the ability to produce antibodies of different specificity? Because the B cells, if you think about it, right, in a person, right, in an individual, all the cells are genetically identical, right? So why would the B cells, which are supposed to be genetically identical, right, would be able to produce different proteins, right? Would be able to produce antibodies that have different antigen binding site. Okay, so this would be the focus for the lecture today, or for this lecture. Alright, so before we look at how, right, we look at the genes that codes for the antibody. So we call that an antibody molecule is a quaternary protein. It consists of light chain and heavy chain, right? Two light chain polypeptides and two heavy chain polypeptides. Okay, so for the light chain, right, it is coded for by two genes, or rather, in our cells, there are two genes that can code for light chain. At any one time for an individual cell, then one of the gene will be expressed to make one kind of light chain polypeptide. All right. So the two genes are found on either chromosome 2 or chromosome 22, and in each of the gene, right, we call it gene clusters, because it's not just one gene; it's really a clusters of genes that together code for one polypeptide. All right, so for each of the gene clusters, there are a series of V genes, the variable genes, followed by a series of J genes or joining genes, and also one or more C genes, the constant genes. Okay, and for each of the V gene, right, there would be a L exon that will code for a leader or signal peptide sequence. Okay, we'll talk a bit more about this when we go into the protein synthesis. Okay, so just remember that for the light chain genes, regardless of whether it's the one on chromosome 2 or chromosome 22, they are actually a clusters of genes, right? You have the V genes, the J genes, and the C genes. Okay, so VJC, yeah, pretty easy to remember, right? Then for the heavy chain, there is only one gene that codes for the heavy chain polypeptide, and it's also a clusters of genes. So we also have the V genes. Similarly, it has a L exon that codes for the leader or signals peptide. There is this additional D gene that is not found in the light chain gene clusters. After that, there's similarly a series of J genes and a series of C genes. Okay, so this VDJC for the heavy chain gene clusters. Okay, so the development of the B cells, right, when they are in the bone marrow, they develop into naive B cells, and in the process, they form or they express the B cell receptor or the membrane bound antibody that serves as B cell receptor. So different B cells, different developing B cells would express a different membrane bound antibody, as we have talked about in the first slide, right? with different specificity. So the process that allows for the expression of different antibodies is known as somatic recombination, or VJ or VDJ recombination. So it would involve the V genes, the D genes, the J genes, right, found in our light chain gene clusters as well as the heavy chain gene clusters. Okay, so for the light chain first, for each of the light chain, right, in terms of the polypeptide, as we have looked at in the previous lecture, when we look at the antibody structure, right, the light chain polypeptide has got two domains: the variable domain that make up part of the antigen binding site, and also the constant domain. Okay, so for the variable domain of the light chain polypeptide, it is coded for by just one of the V genes and one of the J genes. So just now we have looked at the gene clusters for light chain, right, and we saw that it consists of a series of V gene and a series of J gene. But in the end, right, after somatic recombination, each B cell would be left with just one of the V gene and one of the J gene to form or to code for the variable domain of the light chain polypeptide. So we will look at how that is done later. Okay, for the heavy chain is quite similar, right? So the gene cluster coding for the heavy chain has a series of V and series of D and a series of J genes, right? So for each of the B cell, right, during their development, somatic recombination will occur such that they are left with only one of the V and one of the D and one of the J gene, which then later codes for the variable region of the heavy chain. Okay, so this is known as somatic recombination, and because the selection, right, the combination of whether it's VJ for the light chain or VDJ for the heavy chain, it is random, right? So different B cell will have a different combination of VJ and VDJ genes, so they can express different variable regions for the light chain and different variable region for the heavy chain. And the two variable regions, light chain and heavy chain, together form the antigen binding site. So when they have different light chain variable region and different heavy chain variable region or domain, right, then they can have different antigen binding sites between different B cells. Okay, so looking at how the rearrangement, how the different combinations of these genes is achieved, right? So let's focus on the light chain gene cluster first. Okay, so remember again, right, the light chain gene cluster, regardless of whether it's the one on chromosome 2 or chromosome 22, each of them have a series of V genes and a series of J genes. Okay, so somatic recombination would occur, which is catalyzed by the enzyme recombinase, which will randomly select one of the V gene and one of the J genes and combine them together by cutting the DNA between this V gene and J gene. Okay, so when the DNA between this randomly chosen V gene and randomly chosen J gene is cut, and then the V gene and J gene are then joined together, then we have a random combination of what we call a VJ gene pair or a VJ gene segment. Okay. After that, the DNA now for the light chain gene cluster would be a bit shorter, right? Because some of the DNA gets cut away and removed. So the light chain gene cluster is now consisting of a L exon followed by a short intron sequence in the DNA. Then we have this randomly combined VJ gene segment followed by another intron and the one or series of C genes. Okay, there's a promoter in front, obviously, and the terminator at the back, right, at the end of the light chain gene cluster. Okay, so transcription will occur beginning at the L exon, right, and all the way until the terminator sequence downstream of the C genes. After that, at the mRNA level, right, the introns are excised, right? So that would remove the exons are spliced and introns are excised. Sorry, wrong speaking there. Okay, so anyway, after transcription, right, we would have the L exon, intron, VJ mRNA, right, intron, and the C mRNA, right, the C exons. Okay, so the introns would be removed, which means that the intron between the VJ exons, right, and the C exons would be removed, right? So the C exons would be joined to the VJ exon. Okay, so now you have this mature mRNA which contains the L exon, the VJ exons, and the C exon. Okay, so after translation, the L exon will code for the signal peptide, right, or the leader sequence, which will then guide the translating polypeptide, the elongating polypeptide, into the lumen of the rough ER. After which, then the leader sequence is cleaved or cut away. Then the rest of the polypeptide would continue translation, right? So the VJ part would be translated into the variable region of the light chain polypeptide. The C exons, the C part, would be translated into the constant region of the light chain polypeptide. Okay, so a diagram to basically show what I've been talking about. All right, so I'll spend a bit more time here, yeah, because it's a bit abstract. So using the diagram here should make it clearer. All right, so the diagram here shows the template strand of the light chain gene clusters that's found on chromosome 2, right? So I'm not showing the long template strand, so that's just to make it simpler. I'll just show you one strand that is transcribed, the template strand. So again, remember, right, the light chain gene clusters has a series of V genes. So here shows about 40 V genes, a series of J genes. So here shows 5, then 1 or more C genes, right? So happen this one, the one on the chromosome 2 only has 1 C gene. Okay, so somatic recombination occur, right? And it occurs at the DNA level. So we are still at the DNA level, yeah? Somatic recombination occurs, right? Recombinase enzyme would cut away, right? Remove the DNA between a randomly chosen V gene and a randomly chosen J gene. Okay, so in this case, right, the V2, the second V gene is chosen, and the first J gene is chosen, right? So all the DNA in between, right, the V3, V4, V5, all the way to V40, and some of the long coding DNA, right, would be removed. So our V2, the second V gene, and the first J gene, I think it's a typo here, should be J1, right? The first J gene would be combined together, right? JK1, 1, right? So J1, okay? So anyway, we have now this VJ gene pair, right? Or VJ gene segment that is formed in the DNA at the DNA level. Okay, so now transcription would occur, right? So transcription would begin at the L exon of the VJ pair, right? So there will be some control of transcription. The RNA polymerase would know not to transcribe whatever V genes that comes before the VJ gene pair, right? So transcription would begin at the L exon, the DNA sequence for the L exon for the VJ gene pair. So transcription begins here. That's why the mRNA kind of begins here. All right. So the mRNA is transcribed by copying this this part, yeah, of the template strand all the way to the terminator sequence downstream of the C gene. Right, so the mRNA would somewhat begins at the L exon all the way to the C exon that is transcribed from the C gene. Right, then after that, splicing occurs. The post-transcriptional modification occurs with splicing. So the in between of the mRNA, right, would be removed. Okay, so this whole thing gets removed, so our VJ exon now, because it's the mRNA level, right? So the VJ exons would be spliced together with the C exon. So you have this mature mRNA which only contains one V exon, one J exon, and one C exon. All right. So the corresponding mRNA for one V gene, one J gene, and one C gene. Okay. So when this mRNA, this mature mRNA is translated, right, we will have the leader sequence or the signal peptide from the L exon, then the VJ exons together would code for the variable region of the light chain, and the C exon here would code for the, or will be translated into the constant region of the light chain. Is that okay? All right. Okay. Yeah, so if you need to repeat this part, again, this one is a bit abstract, yeah, because it has many different genes, and you need to be clear when we are mentioning DNA level genes and when we are mentioning mRNA level. All right, so some of the wordings used, sometimes I use genes, which is for the DNA level. Sometimes I use exons, which is for the mRNA level. So we have VJ gene, but then at the mRNA level, I will talk about VJ exons. Right, so some change of terms here, so I hope you understand, right, and not be too distracted or bothered by the terms. Okay, so if you need to repeat yourself, need to repeat this part of the video, please go ahead. Right. Yep, so make sure you can understand this part, because the same thing would happen for the heavy chain. Yeah, so if you want understand one part, you understand the whole thing. If you don't, then the whole thing you have problem. Yeah, so make sure you can understand really this somatic recombination for the light chain. Okay, so if you are okay, then I'm just gonna refresh your memory on protein synthesis. So we mentioned the signal peptide, which is coded for by the L exon. Right, so again, we mentioned that the signal peptide or leader sequence, right, function to guide the polypeptide into the denouement of the rough ER. So this is something that we have looked at when we talked about protein synthesis last year. So I hope you recall that. This is the same diagram that I had in my notes and in the lecture as well. Yeah. Okay, so you can see how the signal peptide, right, is removed or cleaved from the rest of the polypeptide after the polypeptide has entered the denouement of the rough ER. Right, so this translated polypeptide would be the light chain polypeptide. Okay, all right. If that's okay, then we would move on to the heavy chain. Right, the same thing kind of happened, but then for the heavy chain, we have this extra series of D genes. Right, so in terms of this random combination of genes or somatic recombination, it has one more step. All right, so the first step that will happen is the somatic recombination, the random recombination of one of the D genes with one of the J genes. So we first form the DJ pair or the DJ gene segment. Okay, and then this DJ gene segment is recombined with one of the V genes. So we have this random combination of VDJ gene segment. Okay, so the mechanism, the process is actually the same. It's by the same enzyme. So there will be the cutting, the removal of DNA in between the chosen genes. It's just that in this case, instead of just VJ, one step, right, we have two steps, DJ first, followed by VDJ. Okay. Alright, so one that is done at the DNA level with the somatic recombination, right, then we would have transcription to form a mRNA, right, the pre-mRNA that contains the VDJ exons in this case, and introns and the C exons as well. So remember, there's also the L exon before the VDJ. Yep. Okay, so after splicing, it's quite similar to what happened with the light chain, right? The C exons would be joined to the VDJ exons. Okay, so now we have a very nice mature mRNA that has that one L exon, the VDJC exons, right? So after translation, then we will have the polypeptide starting with the leader sequence, followed by the variable domain of the heavy chain, which is coded for by the VDJ exons, and then the constant domain of the heavy chain polypeptide, which is coded for by the C exons. All right. So again, another diagram to show, right? So again, this is the template strand. So we have about 200, a series of V genes, right? So about 200. 20 D genes and 6 J genes, followed by 9 C genes, right? So series of V, series of D, series of J, and series of C, right? So somatic recombination occurs first to pair up the D and J. So here, randomly, the first D gene, D1 is chosen. Randomly, the last J is chosen, right? So J6. So the DNA in between are all cut and removed by recombinase, so we get this DJ gene pair. After that, same thing occurs with a randomly chosen V gene, right? So here, the second V gene, L2, right, is chosen, or VH2, V2 is chosen, all right? So the second V gene is now joined to the DJ gene pair to form this randomly chosen VDJ gene segment. Okay, VDJ gene segment. So this would be the end of somatic recombination at the DNA level. After that, transcription would begin at the L exon of the VDJ gene segment. So whatever that's in front would not be transcribed. Right? Transcription begins here. So we have the L exon followed by the VDJ exons because it's mRNA now. Then in between, very long introns, right? Followed by all the C exons, okay? Which they don't show everything lah. They just show 2 here for representation. All right. So after that, splicing occur, right? So the in between of the introns, right, between the C exon and the VDJ exons are removed. So now we have the L exon, VDJ, C exons. Okay, and of course, during splicing, there can be alternative splicing. So this diagram is showing you alternative splicing, but we won't go into that. That is too much details already. So for our lecture, for you, right, I think the most important thing is for you to understand the somatic recombination at the DNA level, followed by how the mature mRNA is formed to join the VDJC. Okay, so that the mature mRNA would be translated, right? So we have the leader sequence, which would be removed from the heavy chain polypeptide. The VDJ again forms the heavy chain variable domain, and the C exons here would be translated into the constant domain of the heavy chain. Okay, so because this somatic recombination is random, right? Random, yeah. So different B cells would form different heavy chain polypeptides and light chain polypeptides. They all differ at the variable region, right? Because the variable region is coded for by different combinations of VJ for light chain and VDJ for heavy chain. Okay? So once the particular B cell has synthesized the light chain and heavy chain polypeptides at the roughly R, then the whole protein synthesis process would continue. The polypeptides would be transported to the Golgi apparatus via the transport vesicle. So we call all the organelles, the protein synthesis that you have learned last year. Okay, so at the Golgi apparatus, that's where the assembly of the polypeptides to form antibody molecules would occur. There would be the formation of the disulfide bonds between the light chains and heavy chains, and there will also be some glycosylation, the addition of carbohydrates, right? And then the fully assembled antibodies would then be transported to the cell surface membrane to be embedded as membrane-bound antibodies serving as B cells receptors if it is a naive B cell, or the antibodies will be secreted if it is already a differentiated plasma cells after the immune activation of the naive B cells. Okay, right, so it's pretty difficult, this part, somatic recombination. So if you need to, do repeat the video and try to understand the process slowly, right? Because it's again pretty abstract, and it happens at the DNA level for somatic recombination, but also involve a bit of splicing in the mRNA level, right? So at different levels, it makes the whole process a little bit more complex. Okay, so do take the time to understand.